Johns Hopkins scientists grow miniature human brains that spark, connect, and mimic real development. In a major leap forward for neuroscience and regenerative biology, researchers at Johns Hopkins University have created an unprecedented multi-region brain organoid, a lab-grown, miniature model of the human brain that mimics real brain structure and activity more closely than ever before. This tiny organoid is not just a cluster of cells. It includes distinct brain regions, rudimentary blood vessels, and most importantly, functional neural networks that fire with electrical activity, similar to what happens in a developing human brain. Scientists say it could dramatically improve how we study complex brain disorders like schizophrenia, autism, and Alzheimer's, offering a window into the early stages of brain development and disease formation. A living model of the whole brain. Traditional brain organoids usually focus on a single brain area, like the cortex or midbrain, but Dr. Annie Kathuria, a biomedical engineer at Johns Hopkins and lead author of the new study, wanted to go further. We've made the next generation of brain organoids, Kathuria explains. Instead of one brain region, we've created a multi-region brain organoid, MRBO, a rudimentary but functional model of the whole brain. This groundbreaking model combines tissues from multiple brain regions and integrates them with endothelial cells that mimic early blood vessel formation. Using light sheet microscopy, the team confirmed the presence of cerebral, midbrain, and hindbrain regions fused together and communicating. How they built it. The scientists started by growing different brain region organoids and primitive blood vessel cells in separate dishes. Once mature enough, they brought them together using bioadhesive proteins, molecular superglue that helped the tissues merge. As the regions connected, something extraordinary happened. Neural networks began to form and communicate, generating spontaneous electrical signals, a sign of emerging brain-like activity. The resulting mini-brain resembled the architecture and behavior of a 40-day-old human fetal brain, with about 6 to 7 million neurons, a fraction of the 86 billion in a fully developed human brain, but enough to demonstrate key developmental processes. Strikingly, around 80% of the cell types typically seen in early human brain development were present, making it one of the most complete lab-grown brain models to date. Why this matters, from brain disorders to drug testing. One of the biggest challenges in neuroscience is understanding what goes wrong in the developing brain that leads to conditions like autism or schizophrenia. Until now, researchers had to rely heavily on animal models, which don't fully capture the complexity of human brain development, especially for psychiatric and neurodevelopmental disorders. You can't just ask someone to let you study their brain cells as they grow, says Kathuria. But with organoids like these, we can now observe disorders unfold in real time, test treatments, and potentially personalize therapies. This breakthrough also addresses a long-standing issue in pharmaceutical development: the high failure rate of neuropsychiatric drugs. Up to 96% of these drugs fail before or during early clinical trials, often because they work in animals but not in humans. Using organoids derived from human cells, researchers hope to test drug candidates in more realistic systems, improving success rates and reducing risks. Beyond the brain, a glimpse into the future of biomedicine. Another exciting aspect of the MRBO is the formation of an early blood-brain barrier, BBB, a critical structure that protects the brain by controlling which substances can enter from the bloodstream. Being able to study how this barrier forms and functions could aid research into stroke, inflammation, and neurodegeneration. Kathuria's team envisions these organoids as platforms for understanding early-onset brain disorders, identifying novel drug targets, testing therapeutic compounds, exploring brain-environment interactions at the cellular level. Conclusion: a powerful tool for studying the human mind. The creation of a functional, multi-region brain organoid represents a major advancement in developmental biology, neuroscience, and personalized medicine. While these organoids are far from conscious or sentient, they provide an ethically viable, scientifically powerful way to study the human brain as it builds itself, one cell at a time. As Kathuria puts it, diseases like autism and schizophrenia don't affect just one part of the brain, they affect the whole thing. Now we have a way to study that whole brain